This is Audiommunity, a podcast about our body's never-ending fight with the outside world. This is Audiommunity episode 10, recorded January 15th, 2015. I'm Kevin Bonham, and joining me from Emory University in Georgia, Matt Woodruff.
Hey, everybody. Happy New Year, 15 days late as of the recording of this.
Matt, since the recording of our last episode, has taken a postdoc position in Emory in Bali Palendran's lab and is doing fun things, but he's also the reason that we haven't recorded anything. It's totally his fault.
Yeah, so last time we recorded,
nothing.
I got a PhD, moved 3 times, moved to Atlanta to start a new job, bought a house, and got engaged.
Wow, I didn't know that. Congratulations.
Yeah, thanks.
I heard about the house from my rotation student.
Who's your rotation student?
Well, I guess he's not mine. He just sits next to me.
No, he's yours. Let's be honest. Kate.
His name is
Charlie.
He's the first year in the immunology program.
Yeah, I know Charlie.
That other lovely voice you hear is Kate Franz. She's still in Boston.
Yes.
Hi, Kate.
Hello. And I just got back from the dentist like 2 hours ago. So my lovely enunciation is probably worse than it normally is, which is saying something.
Yeah, it's normally pretty bad.
Yeah.
So this is the first episode of 2015, and if you are actually still subscribed to this podcast since the last time we put out an episode,
thank you.
You may have noticed that it's been thank you. You may have noticed that it's been a while. Part of it's because Matt has been way too busy. Also, in the meantime, I got married and was teaching like 3 classes and doing a bunch of other stuff. So we're trying something a little bit different this year. We're going to have a little bit of a different format. So as I described in the mini-sode that I posted last week, we are going to record one main episode, of which this is one, per month. And then each of us is going to record a little mini-sode about something. And I guess we're all going to be doing probably our own personal take on things. Kate has promised that she's going to talk mostly about TV and Twilight. But
should be awesome, by the way.
Mm-hmm.
Yeah, I still want to know if vampires can get influenza.
I know.
But we're going to do these... Long pause. We're still going to do these larger episodes once a month, but we're going to try to limit the size of them a little bit. So we're going to try to record for just an hour, not do a whole lot of post-processing, which I was doing before. And so hopefully that means we're going to be more consistent and get more episodes out in a timely fashion.
Yeah, and if we find that we're just more confusing and rushed, then we'll make changes accordingly, I'm sure.
Yeah, hopefully we can make corrections in the mini-sodes if we screw things up too badly.
Right.
Yes.
So with that in mind, let's jump straight into the topic for today's episode, which is a paper that was published recently in the Journal of Experimental Medicine called Staphylococcus aureus infection induces protein A-mediated immune evasion in humans. And this is from a lab at the University of Chicago, Noel Pauli, first author, and Patrick Wilson, last author.
Can I say something about the authors while we're on the topic? I really like the author list because one of the authors is like a bizarro me. Like her name is almost my name, but not quite. Karen M. Frank. It's almost like Kate M. Franz. That is my scientific contribution. You are welcome.
Thank you very much.
Drop the mic. We're done here.
We're totally going to make it under 60 minutes.
Thanks, bizarro me. And out.
Oh, you guys are really challenging my resolution to not do any editing of this podcast.
No, I don't think that needs to be edited at all.
It's what the people want. You got to give the people what they want.
Right.
Okay. Fair enough.
Kate want bizarro Kate.
Yeah, everybody wants Bizarre Kate. So before we get into the meat of the paper, and I gather from our conversations before we started recording, Matt has some issues with the meat of the paper. But before we get to that, I want to give a little bit of background. So this paper is about Staph aureus. And even though we're not a microbiology podcast, I thought it was worth talking a little bit about this bug just to put it in context. So Staph aureus is a gram-positive bacterium, and it causes most of what we commonly refer to as staph infections, which kind of makes sense. But staph infections are a group of a lot of different Staphylococcus-mediated infections. Staph aureus is the most common cause of staph infections, but it's not always an infectious microbe. So actually, Staph aureus is commensal. In other words, it's part of the normal flora on about 20% of people worldwide. So it's all over the place. But if it gets into a wound, or if it gets into an area where it has access to parts of the body that it maybe shouldn't, then it can cause cause a pretty nasty infection.
So, Kevin, where does Staph aureus normally live? If it's
it normally lives on the skin.
Yes, that is correct, sir.
Correct.
Oh, were you quizzing me? I thought you were actually asking.
So, I mean, I don't want to always bring it to this place, but I will. This is a topic that was gone over in Grey's Anatomy last season with Bailey. She had staph infection just on her skin, and she's a surgeon, and she was surgically manipulating someone's body, and they got a staph infection from her skin in a hospital. It's very common. It's just on your hands, in your nose.
Yeah, and the nape of your nose,
and
right?
Killing
I think it
people in Grey's Anatomy.
Every time.
That's why surgeons are supposed to scrub in. And yeah, so actually, it is a major cause of what we call nosocomial infections or hospital-acquired infections. In particular, there are a couple of really, really bad versions of Staph aureus that some of you might have heard of. One of them is called MRSA, MRSA, which is methicillin-resistant Staph aureus, and a sort of newer form, which is VRSA, or vancomycin-resistant Staph aureus. Basically, these are versions of Staph aureus or strains of Staph aureus that have resistance to some of the most powerful antibiotics that we have available. So if you get these infections, they're very hard to get rid of, and they can cause serious damage. And so a lot of people who get hospital-acquired infections have MRSA or VRSA, and they can be really problematic, and they're really expensive to treat and cause all sorts of other problems. But I did want to mention there are a couple of other versions of staph that cause infections sometimes. So one of them is Staphylococcus epidermidis, and that is another normal skin commensal that's present on a lot of people. But if you're immunocompromised or if you get a wound, you can get epidermidis infections. And then another one is Staph saprophyticus, which is a genital commensal. So it's normally found in why did my brain just blank?
In the genitals?
In the genitals. Yes,
that's what I heard too.
Okay, let's try this again. So there's also Staph saprophyticus, which is a normal commensal of the genital tract and is the cause of some urogenital infections. Or
like a UTI?
UTI, yes. Yeah. I don't know why my brain isn't functioning right now. I'm going to edit a bunch of this.
It's the beer.
It's probably because of the beer.
I've only had one. It doesn't affect me that bad. Okay. But we're not talking about those other types of staph. We're talking about, what are you doing, Kate?
I drew a picture. Sorry.
Can't really see it.
It's like a bodybuilder with an afro. Okay, anyway.
Does he have staph?
No.
Okay. Staph-free bodybuilder.
Yeah, go ahead.
Cool. So staph causes infection in a number of different ways. Like I said, it can get into wounds and it can cause septicemia. It's also the cause of a lot of versions of what is commonly called flesh-eating disease, although that's not quite an accurate term. But one of the things that Staph aureus does is it produces a number of toxins that can cause all kinds of reactions in different cell types. And in particular, I wanted to mention something called toxic shock syndrome, which was...
Oh, your baby.
What's that?
I said, oh, your baby. Your project. What? That never was.
That wasn't my project.
Really?
That was endotoxin tolerance. Endotoxin tolerance is different.
Oh, toxic shock syndrome, like the tampons. Okay, sorry.
Yeah, like tampons, exactly. So this
I was
is a thing...
like 30 seconds behind on the septicemia.
Oh, okay. Fair enough. So toxic shock syndrome is something that if you are older than the three of us, you might actually remember. This was a thing that happened in the 1970s. And basically, when tampon producers started to make tampons that were super absorbent and could stay in the body for a really long period of time without being removed, a bunch of people started to come down with staph-mediated toxic shock syndrome. And it was kind of a mystery what was causing it for a long time. But basically, the end result is that Staph aureus that was present probably on the hands of the people that were inserting the tampons got into the vagina through the tampon and then started to produce this toxic shock syndrome toxin, which is something that we've briefly mentioned in previous podcasts. It's called a super antigen.
Wasn't it also that the tampons were so absorbent they were drying out the tract, and so when you removed them, you'd get micro tears, and then you would get the bacteria inside of you?
That sounds correct. Although, strangely, when I was teaching about this past semester in my infectious disease class, it turned out that a large number of toxic shock syndrome cases were actually in boys. So, unclear exactly why that was happening. But regardless, it was linked to tampons.
What? Like
at
in
the time.
Oh, you know what
Not
it is?
for the boys, no. In general, people normally associate it with tampons.
I mean,
I thought it was going to be like...
We're not judgmental.
I thought it was going to be like wrestlers or athletes, like they put the tampons up their nose when they get a bloody nose. And then they were like,
oh,
and they already have like
Yeah.
staph in their nose, and then they pull it out. But the taco shock in men, I'm going to say it's probably separate. It's not
The taco shock?
Tacos. I go into taco shock every Tuesday.
I can't.
Taco
I'm half numb.
Tuesday.
The toxic shock.
Thank you. Okay.
welcome.
So
You should read The Coming Plague by Lori Garrett to learn all about this.
you
It's
should.
a
It's
lovely,
great.
lovely book.
That was a textbook I used in my class, actually, because it's awesome. In any case, TSST, toxic shock, I can't even say it. Toxic shock
taco shock.
syndrome toxin. Taco shock syndrome toxin. That's much easier to say.
That is.
I think you get that when
didn't
you
even go
have
to the
too many
dentist today.
Dorito Locos tacos.
So one.
This is what it's like, people. You have not been subjected to this before. This is what it's like when we don't edit the podcast.
It's better this way. Got to give the people. I'm stealing that from another podcast. Shout out to Jalen Rose. Give the people what they want. Okay, continue.
That was lovely.
You're welcome.
Kate, by the way, small aside, Kate did some masterful karaoke at my wedding.
Yeah,
That's
she
right.
killed
Pretty
it.
wonderful.
That's right.
She killed it. It was good.
What is his name? John, my boss. Okay. John was like, we need to go karaoke for the holiday party. Like, that was really impressive. And I was like, yeah, we should definitely go karaoke, John, because you're so good at karaoke too. Yeah. He was awful. It was impressively bad.
You know that I'm not editing this part out, right, Kate?
Oh, you think John's ever going to listen to this?
Oops.
That's a good point.
Fair point.
John doesn't
Okay.
even know how to work his own phone to call someone. He can't get on the internet. Sorry. Edit that
Coming
part out.
back.
Edit that part out.
I won't. Getting back to Staph aureus, toxic shock syndrome toxin is a super antigen.
Getting
And as we've
fired.
mentioned before, what a super antigen does is it essentially activates the immune system. And you might think this is a weird thing for a microbe to do. Why would you want to activate the immune system if you're trying to cause an infection? But the key part is they are non-specifically activating the cells of the adaptive immune system, like T cells and B cells. And what this means is that, in general, less than 1%, usually less than 0.1% of the T cells and B cells in the body are going to be specific for any one antigen. And part of what the immune system does is during an infection, the B cells and T cells that are specific for the antigen will undergo activation-induced replication. So they'll make a bunch of copies of themselves. So during the infection, most of the T cells and B cells in the body that are present are ones that are specific because those have replicated. When you do non-specific activation of the immune system, basically what's happening is all of the immune system, all of the adaptive cells of the immune system are getting activated. So that tiny little percentage that is actually antigen specific has to compete with all of the non-specific ones that can't actually target the bug, and therefore the bug is sort of protected. So you don't get the same adaptive response. I'm not explaining this very well.
Basically, you bias all the responses to be against something that doesn't matter, and so it hides. It can hide or prevent like a productive response from developing.
Right, exactly. So there's like cytokines and growth factors and all of these things that T cells and B cells need. And if the actual antigen-specific ones have to compete with all of these other non-specific cells, you're not going to get as robust a response to the bug itself. And so I'm talking about...
Sorry, I'm going to
ahead.
jump in here real quick. So one of the interesting logical outcomes of that is that there is a fixed capacity. There's a fixed capacity of the adaptive immune system, right? In order for that to make any sense, it means that the adaptive immune system can respond to a lot of things simultaneously, but there is certainly a limit to that. And what's interesting, I think, about this paper is it brings up that question, like what actually is the capacity of the adaptive immune system? How many different antigens can you respond to at the same time? Unfortunately, this isn't the sort of paper that tries to dig down into that mechanism, but I do think it's an interesting concept, and we'll talk more about that, I'm sure, as we get into it.
So are you saying that if you just expose yourself to a bunch of antigens, you could prevent something like an autoimmune disease? Because you would use up the capacity of the immune system to respond while it's responding to all these other antigens, and it will stop like responding to crap inside of it. You just can flood out the response.
So I think the there are a number of different problems with autoimmunity. One is that antigen is constantly, like new antigen is constantly being produced. So what you would need is a constant flooding of that site with random protein.
If you knew
I think it would be
precisely
hard.
if you knew precisely when you are gonna get the stimulus that activated that autoreactive cell, you might be able to do something about it. But generally speaking, we don't know. Like, once you are diagnosed with MS, you already have a crazy immune response going against myelin. So at that stage, you're probably not going to be able to outcompete it. I think one of the sort of hand-wavy explanations around the hygiene hypothesis, which is this notion that people who are exposed to more microbes in general, people who are in less sort of hygienic or antiseptic environments are less prone to allergies and autoimmunity, is precisely this notion that if the immune system is being exposed to things that are actually threats more often, you are less likely to develop a response against something that you're not supposed to, because usually the antigenic stimulation for an autoimmune disorder is weak, but it's just strong enough that it can start the ball rolling, and then you get these positive feedback loops.
Yeah, and I think there's more complexity built into that as well, as you get into regulation and whether that's an actual threshold of response or whether you've modified the local environments such that barriers aren't exactly the same. So I think there's a lot of things that could be playing into that.
Yeah, for sure. So anyway, I bring up this TSST, toxic shock syndrome toxin, because it's a super antigen, and that becomes important for this paper where they're talking about another different Staph aureus produced super antigen. And whereas TSST targets T cells and T cell MHC interaction, this protein that we're going to be talking about in this paper is called protein A, which is not a particularly creative name, abbreviated SPA in staph. And this SPA is a super antigen, but it's a super antigen for the B cell receptor. So in other words, it binds non-specifically to B cell receptors, which, if you remember, are the basically membrane-bound portion of an antibody, or membrane-bound version of an antibody, rather. So SPA binds to that and non-specifically activates B cells. And it can actually bind in two different places. There are a couple of different isoforms of SPA, one of which binds to the constant region, the FC domain of IgG, which is one of these isotypes of antibodies, and it's the constant region. So if you imagine the Y of an antibody, it's the base of the antibody. And then another portion of SPA binds to what's called VH3. So this is one of the heavy chain variants that can be produced during RAG recombination of the B cell receptor. So just want to give a quick review. We've talked about it in previous episodes, but B cell receptors are generated through the rearrangement, the VDJ recombination of antibody genes. And part of that VDJ is selecting 1V, 1D, and 1J for the chains of the antibody. And so VH3 is just one of the V versions that is found in a lot of human B cell receptors. But the key fact here is that even though this superantigen is binding to VH3, it is non-specifically binding to VH3. So in other words, the antigen binding capacity of the antibody is not what's at stake here. It's sort of binding to the outside portion of the antibody, not to the normal antigen binding site of the antibody. Hopefully that makes sense. If it doesn't, maybe we need to draw a picture. But SPA is, they say in the paper that it binds to about 30% of all B cells in the body, as opposed to, as I said, the less than 1%, sometimes a tenth or 100% of the B cells that normally respond to any given antigen. So the question that they're asking in this paper is, well, let me go back for just a sec. So what we know prior to this paper is that Staph aureus can cause infection, and you get some B cell responses to Staph aureus infection. So you can find antibodies to various staph components in the blood of people who have previously been infected with staph. But those antibodies do not seem to confer any protection against future staph infection. Okay, so
So they do say that in the paper.
Yeah.
I want to point out that while that was true for a mortality curve, what they did show is that it did actually decrease morbidity.
Okay.
So there was an effect of having those antibodies around. And, you mentioned that there were some issues that I had with the paper. I think actually the paper was fine for the most part, but there are just some concepts that I think get a little confusing as far as what they think the antibodies are actually doing, what they're actually targeting. So I think that it's true, clearly, that the antibodies are not, you're not going to get super productive vaccination out of them. But I think to go so far as to say that they're not useful is probably an overstatement.
Right. So maybe let's say it's not like you get infected with influenza and then you're immune from that strain of influenza.
Correct.
Maybe the antibody-mediated response provides some protection or it reduces the symptoms of disease, but it's not going to make you immune. You don't get protective immunity.
Right.
From an infection.
I was actually just confused. How did they even draw that conclusion to begin with? That the antibodies didn't matter.
So they cite a couple different... Hold on, let me think before I start talking. I think it was based largely on previous studies that had shown that first of all, people get recurring infections to staph. So if you assume that the antibodies are fine and you can find antibodies in the blood, you shouldn't have people that get these chronic and recurring infections. But I think they also cite a couple papers that show that vaccine attempts against Staph aureus, basically you can get a productive antibody response, but it doesn't really turn into anything. It doesn't actually mediate protection.
Okay.
So it's productive but not protective.
Right. Yeah, you're getting something out. Although they do cite this one paper where they have a transgenic mouse where all the B cells can bind to SPA. So it's basically a V3 heavy chain knock-in. We'll talk about that in a second. But basically all the B cells in the entire mouse can all bind to this protein. And what they find is when you inject this protein, all of those B cells die. And so they sort of assume that that's the normal mechanism of how staph prevents response in mice, is that it just kills all of the specific B cells with this protein, which is, clearly not happening in humans. I would say, though, that if you've got a mouse where every single B cell looks the same and you're overstimulating all of them at the same time, I could see a situation where just the fact that they're all competing for attention, basically, from all those survival signals that you were talking about would lead to a mass extinction rather than, an activation. So right. I'm not convinced that that's a reasonable conclusion.
All right, fair enough. But so the point of this paper, the reason that they say that they're starting it is because there are B cells that are trying to produce antibodies in people. And we know that SPA has some effect, or they can find antibodies that are against SPA. And so the hypothesis, because SPA is the superantigen and can bind to the B cell receptor, they want to answer the hypothesis of whether the Staph aureus can evade the immune system by activating B cells through this SPA. So that's the sort of hypothesis that's driving this paper. Matt, your hair looks ridiculous, by the way.
Yeah, that's what happens when you wear hats. I actually went home and took a nap today because I was up at 5 I was up at 4 in the lab by 5 because I needed to do a sort by 10. So I decided it was reasonable for me to go home for 2 hours and take a nap. And then I woke up and I was like, oh, it's very clear that I took a nap. So then I put on a hat. That's the story of my hair.
Excellent. Maybe we should take a screen cap and then post that on the website.
Oh, that's so mean.
It's fine.
Okay, so what these people did is they basically went and took patients that had ongoing staph infections. So they took blood from 18 patients, 15 of which had a current staph infection, and 3 of which had a staph infection but also had chronic staph infections. So they've been coming to the hospital multiple times with staph infections. And they essentially tested the serum of the patients to look for serological responses, or essentially responses that are visible in the blood. That's antibodies, essentially. They looked for those in these patients. And basically, the serological responses looked fine. It looked like they had productive immune responses against staph, and both the acutely infected and the chronically infected people had pretty similar serological tests. So they have antibodies to staph present in their blood, and they have sort of normal B cell activation. It's not like they don't see any B cells or they don't see any T cells. They look sort of normal for an infected person.
Yeah, and these are targets that they know are important, right? So these are membrane targets of staph, and so if you produce antibodies against these things, there's a reasonable assumption assumption that, those antibodies should be at some level productive in protection.
Right. So basically, all these patients, they look sort of normal as far as staph infections go. So then what they did is they looked at the plasmablast responses. And I should probably have pointed out, a plasmablast is essentially just an activated B cell. So when B cells get activated, they turn into plasma cells, which are the cells that are secreting boatloads of antibody. And so blasting is essentially replicating. And so you have replicating plasma cells. That's a plasmablast. And importantly, these are the cells that are going to go on to become memory B cells, the long-term memory B cells that are going to give you protective immunity over a lifetime.
Did you just say blasting as a verb?
I did.
Okay, just...
Is that not okay?
No, I'm really into it. Wait, can you re-say how you said blasting as a verb? Like...
Blasting is actually a verb.
The cells are blasting.
Yeah, the cells are blasting?
Yeah.
Mm-hmm. Cells
as
can blast.
They are like in the state of being plasmablasts, or they're blasting antibody out? It's like...
No.
Blasting is dividing. So T cell, when a T cell blasts, it is replicating. When a B cell blasts, it is undergoing activation-induced replication.
I've never heard blasting. This verb. I'm really into it. Why do you guys keep taking pictures? It's freaking creepy.
Me. Because there's a capture thing. Kevin sent a capture thing to take a picture of my hair, and I figured he wouldn't take the picture of me, so I'd have to take the picture of myself.
Thank you for doing that, Matt.
Yep, no problem.
I appreciate it.
And then the first one was accidentally of Kevin because he was up on my screen. So now I have a picture of Kevin and a picture of me with stupid hair.
Make sure you send that to me so I can put it on the website.
Yeah, I'll get it done.
Okay, so sorry for my use of blasting as a verb.
I
Just
like
replication.
it. I've never heard that. It was very interesting. I want to use it like colloquially now. I can't say that word either. Whatever. Don't edit this out.
No, we're going to just remember that Kate went to the dentist recently and her mouth is numb. And we'll just pretend that is the case for every other episode.
Exactly.
As well.
Thank you.
Yeah.
So I before we get off the topic of blasting cells, plasmablasts, I think it's important to know where these plasmablasts are coming from in these patients. So as you mentioned, Kevin, they did pull the serum from these patients to take a look at antibody reactivity against staph. While they were doing that, presumably they also took cells out of the peripheral blood. And so what they're looking at is B cells that have recently become activated. They're in the process of they're going from naive B cell in a germinal center to either a plasma cell, like you said, which is going to pump out antibodies, or a memory B cell. And so it's sort of this weird in-between phase where they're pumping out a little bit of antibody, but they're not quite sure exactly what they're going to be doing yet. And the reason I think that it's interesting is because they are certainly getting plasmablasts out of the blood. I would expect a significant number of the actual plasmablasts involved in the infection to be in the lymph nodes, still undergoing a germinal center response.
Right.
And I think it would be really important, maybe as a follow-up to this study, to know exactly what's going on in the lymph node itself. Unfortunately, with humans, you can't really do that. You can't just pull someone's lymph node like we do with mice and then, test it and stain it and all of those things. So it becomes a little more difficult. But I think this paper could be a lot more powerful with some sort of mouse model that at least allows you to know what's going on in these lymph nodes.
Yeah, that would be nice. But as you say, our window into the human immune system is a little bit more limited, unfortunately. And if the difference in SPA responses in mice and humans is real, it sounds like maybe there's some question about whether it's an artifact of the model. But if there is that difference, then obviously that is not possible, which is an unfortunate limitation of experimenting on people. But probably for the best.
Let's say experimenting with patient samples. That's a better way of putting that.
Oh, fine. In any case, so the next thing that they did, so they have these plasmablasts. They can basically isolate B cells and the antibodies that they're producing. And then they can sequence the B cell receptor locus. So this is the gene that's producing the B cell receptor or antibody. They can sequence it and then look at what it is and infer a lot about what has happened to that individual B cell in the past based on how much the DNA for its B cell receptor is rearranged. Because remember, the changes that are happening to B cells are happening at the DNA level. So we can really simply sequence that genetic locus And C, did it undergo VDJ recombination? What sort of VDJ recombination? Has it undergone somatic hypermutation, which is this process of sort of selectively increasing the affinity of the antibody over time in an immune response? And I think we've talked about that on previous episodes too. But when they isolate these B cells and they look at the BCR locus, at the first step, they're just sort of seeing, do the B cells look normal? In other words, one potential mechanism for the way that SPA could inhibit B cell responses is just to kill B cells, right? You could get rid of all of the B cells that are specific for SPA or that SPA binds to. Sort of like we were talking about in the mouse model, where if a B cell binds to SPA, maybe it gets deleted for some reason. And that's not what they find. What they find is that the BCR loci of the B cells in these patients look pretty normal. They're sort of skewed towards this VH3 locus, which you would expect if SPA is inducing activation of these B cells. They've undergone productive rearrangements and they've undergone this somatic hypermutation. So they have accumulated mutations past the germline version of these genes, suggesting that they've undergone germinal center responses. They've undergone somatic hypermutation. They've been activated the way that a B cell normally would have been activated.
And you mentioned that this, so previous studies, I guess, have shown this, right? That SPA specifically targets using its function as a superantigen. Right. It specifically targets these VH3 variable heavy region 3 positive B cells. And so it turns out that that is the most abundant kind of heavy V region in B cells. So it's present in about 30% of B cells, which is where that 30% of B cells which are responsive to SPA comes in.
Right. So the B cells look normal. The immune systems look, normal for an infection. They have normal serological results. So then the question is, since SPA can bind to VH3, let's look at B cells based on their VH locus. And as I mentioned, they saw significant enrichment for the VH3 antibodies in the B cells that were activated in these patients. So in other words, if you look at the B cells in the patients, you would expect a certain amount of them to have VH3. But if you look at patients that have been infected with staph, essentially that proportion of VH3 B cells goes up relative to people who are infected with influenza as a control. So that's a non-relevant infection, or to patients that don't have an active infection. So you have an enrichment for this VH3, which, as I said, you would expect because the B cells that are activated are replicating. So they're becoming a larger percentage of the total pool.
Yeah. And it'd be really nice, again, if you could see what the naive B cell repertoire looked like in these mice. I think that they mentioned that the naive B cell repertoire was normal, but I don't remember actually seeing the data suggesting that.
Yeah, they actually looked at, I dug into the methods for that because I was a little bit confused about that. They actually are comparing patients that are not infected with Staph aureus. So
right.
They're looking at naive, what they're calling naive are patients that don't have a documented history of staph infection.
Yeah. So it'd be nice if you could go into these patients, look at the normal non-activated B cell repertoire and see, although maybe, I don't know, is this completely systemic? Maybe you can't find VH3s that aren't active in an actively infected person.
Yeah, maybe. Certainly maybe not in the peripheral blood, right?
Yeah, you wouldn't find them in the peripheral blood. Yeah, you'd have to start pulling lymph nodes again, and we've sort of established we're not going to do that.
Yeah. So then the next thing they did is they looked at reactivity to SPA. So we know that we're enriched for VH3. So you can sort of naively assume that you would have increased reactivity to SPA if you have a lot of these VH3 B cells floating around. And that is certainly what they find. But what I thought was kind of interesting is that they actually find that the plasmablasts have affinity matured toward reactivity to SPA. And this data is a little bit, the results aren't huge. There's not like clearly different bar graphs. You really need statistics to actually say that there's a difference. But they do see statistically significant increases in binding. So in other words, during this affinity maturation step where the B cell receptors are mutating and then being selected for increased binding, they're finding mutations in these B cell receptors that cause the antigen binding portion of the antibody to have increased affinity for SPA. In some cases, to the detriment of the super antigenic binding of SPA. And so that's a little bit bit maybe confusing of a statement. Basically, if you think about an antibody as this Y, the tip of one of the arms of the Y is the antigen binding site. That's not what's being bound by the super antigenic portion of SPA. SPA is normally binding to sort of the outside of that arm. But some of these affinity matured B cells are actually having more binding capacity on the antigenic port, the tip of that arm, than on the side at the expense of the binding on the side. So this is really suggesting that during the course of an immune response to SPA, even though SPA is binding to the outside of the antibody normally, the affinity maturation is actually increasing the affinity of the antigen binding portion towards this antigen, irrespective of its super antigenic capacity. That was one of the things that I found actually most interesting in this paper, is that even though you have this other mechanism of binding, the normal affinity maturation is still taking place and actually seems to be doing a fairly good job. And they actually go through and sort of do revertant mutations. They take these antibodies and replace the mutated nucleotides with the germline nucleotides, and they find that they lose that binding. So that was pretty cool.
Yeah, it's interesting. I'm concerned about a number of things. The first thing is, if we want to back up for just a second, figure 1 shows serum responses to all of these different known targets to staph by control versus infected patients. And the result of that is that these patients, these infected patients, are producing antibodies at levels that are high, first of all, seemingly. Although, yeah, it's not a terribly quantitative technique. But regardless, you can detect it. So you are getting antibody production of, all of these different, against all of these different targets. And then if we move to figure 2D, what we find is that none of the cells that are responsible for any of the productive antibody that we just saw in figure 1A are in the peripheral blood, right? None of the blasting population, there you go, Kate. None of the blasting population that they're pulling from the blood is actually responsible for any of the antibody targets or any of the antibody specificities that we were actually looking for in figure 1A. Mm-hmm. And so my question is then, what is the relevance of these plasmablasts in the blood, right? If they're not the ones that are secreting the antibodies that we're picking up, then what are we actually looking at? Where are those cells? And this is why I brought up the lymph node thing. I really want to look inside these lymph nodes, because what if the cells that are actually responsible for clearing this infection, or at least combating this infection, just aren't in the peripheral blood? They just can't find them.
Right.
So there's no question, that you're getting an expansion of these VH3 positive B cells. It's just not clear to me how important that is. I mean, it's an interesting finding, and clearly it must be doing something, right? But
it would be really nice, again, if we had a mouse model, or I don't know if there are clinical isolates that are SPA negative, but what would be really nice is to look at microbes that don't have SPA and compare them to the microbes that do, because maybe those responses that you see to other Staph aureus antigens in figure 1A,
right,
maybe those would be 10 times higher in people that were infected with a protein A negative staph.
So I think that that's an important point, and it brings up another point. And so some of those experiments have been done in mice models. I went back into some of their literature. And so one of the primary sources that they're citing is a study looking exactly at what you're saying, where you're taking staph, and then you're taking staph where you have mutants of SPA, and you're basically just injecting them straight into the blood of mice. And so the basic readout that they were using is, okay, if I take, 10,000 Staph aureus bacteria, right, and I inject it into the blood, and then I wait for however many hours, and then I pull out that blood, how much infective staph is still in the blood? So if the immune system, the idea here is that if the immune system is doing a good job of clearing it, then you'll be able to pick out less infective staph several hours later. And so what they find is that if you basically knock out or attenuate SPA, so you basically block its super antigen capacity, what you get increased clearance of staph from the blood. So clearly, SPA is important in protecting staph from being cleared by the immune system. But what's really interesting about that study is the following observation, which is that they suspected that the superantigen capacity against the antibody was antibody mediated. So the protection of SPA in protecting staph from being cleared actually requires antibody responses against it. And so what they did was they took that same mutant, they basically knocked out the other side of the equation. So they knocked out the antibody side. So they used a mouse model called a MUMT mouse, which basically does not produce antibody. And what they found was that in the complete absence of antibody, staph clearance increased. So basically what that said was SPA is not effective in protecting staph when there's no antibody present in the mouse. So that's interesting, but it brings up a really important point, which is if you don't need antibody to clear staph, then what are we talking about? You know, we sort of start this paper on the assumption that the way that we're going to protect people from this bug, because it's extracellular, is we're going to produce a whole bunch of antibodies like we think about with virtually every vaccination. We're going to produce a whole bunch of antibodies in the blood, and then that's going to get out into the tissue, and that's going to protect us from infection.
Yes. Thank you, Matt. That was my biggest problem with this paper is they just start off talking about how obviously antibodies are important. I was like, why is this an obvious thing? You have there's nothing here that's convincing me that antibodies are the answer. In fact, you just spent a whole paper convincing me they're not.
So are you telling me, Matt, so let me just rephrase the conclusions of that other paper that you're talking about, which, by the way, send me that link so we can put it in the show notes.
Yeah.
If you have... So staph gets into the blood, it's protected from clearance.
Yep.
And that protection is SPA mediated. But if you don't have any B cells, it doesn't matter if the staph has SPA or not, it gets cleared in the absence of B cells.
Correct. That was one of the figures of that paper. Or at least, that was what the conclusions were. So
are you saying then that we should basically be giving patients B cell ablating therapy in order to cure them of staph?
No, absolutely not. Yeah, so what we're going to do in order to combat staph is give everyone type 1 immune deficiencies, primary immune deficiencies, right? So we'll just knock out all the B and T cells, and I'm sure you'll be fine.
You just give them rituximab.
Yeah, I was just going to say that. What about just a short dose?
Yeah, well, that's fine. No, I think, I don't know, it's an interesting thing. And I think the more interesting point, and I mentioned it earlier about, or for me surrounding this paper, is the idea of immunodominance, right? So this paper is trying to show, and I think it does convincingly, that SPA does target this really broad swath of B cells, right? So you're getting nonspecific activation of a huge compartment of the B cell repertoire in humans. And clearly that must be doing something. And I think that probably what you were speculating before was right, was that you do have B cell to B cell competition. And if there's so much competition because everybody's trying to get active all at the same time, it's going to be hard for any one B cell clone to really focus in on anything. But I think the mechanism of how that actually happens is really unclear. And this paper is interesting in that, it observes that all of this is happening, but I think it's making a whole lot of conclusions about how you might get this phenomenon of immunodominance without actually knowing, the real basics.
I agree.
There was one other thing.
Do you think that they kind of propose this mechanism that you have essentially just confused B cells, so everything activates, so you don't get any one great response. But do you think that coupled with the previous paper that there might be something in the B cell activation that's just like pro growth of staph, as opposed to when the B cells are there, they're just confused, they can't see staph, but actually when the B cells are there and they're all blasting that they're doing something that's...
Way to go, Kate. Way to incorporate new vocabulary.
I can't believe I've gone this long without hearing that word. When the B cells are blasting, there's actually something that's like that Staph aureus needs for its life cycle, or like for just to replicate more efficiently. Infection.
Yeah, well, I mean, an interesting point of the last paper, this paper that I keep referencing and we haven't actually talked about, is that
maybe we'll do that on the next episode. Do it on a mini-sode. That's what you should
maybe. Yeah, we could do that on a mini-sode. So in that paper, they suggest that SPA doesn't work without productive antibody response against it, basically, right? Mm-hmm. Or it could also be formally that that the environment without B cells, right, is sufficiently messed up where there's some other problem that staph has.
Yeah.
So
fair.
it's not necessarily true, but it would certainly suggest that SPA has some sort of requirement of antibody response in order to be effective.
Did they do infection? I'm sorry. I think I might have just zoned out for a second because I'm very hungry. Did they do the VH3 negative infections?
Well, no, because this is all in human patients, so you don't have any VH3.
No, the other. Wait, wasn't the other thing in mice?
The other was in mice, and I don't know.
Okay.
I don't know. I'd have to go back and look.
Okay. Anyway.
That would be a good experiment.
Yeah, it would. So I want to bring up one more thing, just because I think the impact of this paper is driven by the idea, and Kevin, you mentioned it at the very beginning, it's driven by the idea that in order to produce an effective antibody-mediated vaccine against Staph aureus, one approach might be to first knock out the ability of SPA to protect staph from antibody response, and then subsequently to vaccinate against staph without SPA. Right. So you're basically eliminating SPA's ability to control your immune response and then injecting staph after that to get an effective vaccine. So that assumes a couple things. That assumes that during primary infection, when you're looking at SPA responses, none of those antibodies are actually shutting down SPA.
Right. Yeah. They don't actually demonstrate whether any antibody binding to SPA is neutralizing. Right?
Exactly.
When we vaccinate against a lot of bacterial infections like tetanus, we're not actually vaccinating against the microbe Clostridium tetani. We're vaccinating against tetanus toxin. And if you can eliminate the ability of the toxin to cause disease, you eliminate the ability of the microbe to cause disease, which is cool. But in this case, because the toxin is targeting antibodies anyway, can you get an antibody that neutralizes, that binds to and prevents the normal activity of the toxin? And that I was unclear about, and it is definitely unclear to me if that's possible.
I don't think that they tested it directly, but if you look at figure 3, for example, in figure 3, you talked about that reversion analysis that they did, where basically they took all of these affinity matured antibodies and they brought them back to their baseline, where they started essentially before they started the immune response.
Right.
And so they randomly picked out 5 antibodies to do this with. I assume it was random. I assume they just picked 5, and these were the 5 that showed up. Of the 5 that showed up, one of them, first of all, was non-VH3. So that means that you're getting B cell responses against SPA that are not targeted by the superantigen function of SPA.
Right.
Which would suggest to me that there's a strong likelihood that those responses are going to be productive because they're not all fouled up by the superantigen binding.
Right.
And more importantly, you can actually see that this binding goes down. So in the non-VH3 target, you've got 2 different mutants of SPA that they're testing against. One is a mutant for the superantigen function that you were talking about, and the other is a mutant for a different function that SPA is also doing, which prevents it from being phagocytosed. But basically what you see is that by reverting, so you're getting binding of non-VH3 antibodies to SPA in a method that is independent of the function of SPA. And so now you have to show me that those antibodies aren't actually inhibiting SPA, because if they are, then the idea that knocking out SPA and then vaccinating is going to give you a better antibody response is completely moot, because that's happening in vivo.
Right.
And further...
Yeah, but we don't know the proportion, right? So
we don't.
It depends on just producing a neutralizing antibody.
Right.
It depends on the quantity that's being produced. If it's only 10% of your total B cell production, then
true.
Maybe that's effective, maybe it's not. But I think that
right, maybe it's low. So let me follow that up with this, and this is largely borrowed from Patrick Schleivert. I hope I'm saying the name right. Who is a faculty member at the University of Iowa, and he brings up several points in a discussion piece about this paper. The first is actually, I was glad that you mentioned toxic shock syndrome. And,
taco shock?
Shock. Exactly. Taco shock. So while it's true that this is a cause, or this is caused by staph, if you look into the blood of about 80% of females, they all have antibodies that render them resistant to that toxin, which means that in those cases, those patients have overcome this mechanism that they're describing in this paper. So there's an 80% success rate in that scenario where you are getting good responses against Staph aureus, and that is sort of largely ignored by this paper. In addition to that, if you look at pooled IVIG, which is what we give to patients with primary immunodeficiencies who can't make their own antibodies. And so this is just immunoglobulins, antibodies pooled from thousands of people, all mashed together and injected into people that can't make antibodies for themselves. There are several antibodies that are effective against staph. And actually, one of the treatments for someone with advanced staph is to give them a ton of IVIG from other patients, which means that other people are making productive antibodies against this bug, right? So this is certainly not a complete shutdown. And I think the last thing that I would say is, unfortunately, he did not provide a reference, and I don't know the field well enough to track it down myself. I'm sure with enough time, but apparently there has been a recent vaccination attempt where they coupled an SPA target antigen with two other important surface antigens of staph, and the trial failed, basically. So they tried to vaccinate against SPA alongside a couple different antigens to see if they could get productive response, and ultimately the trial didn't work. And, certainly there are a number of reasons that the trial might not have worked. It certainly could be that, this mechanism is working the way that they think it is, and, this trial just failed for other reasons. But I don't know. I think that there are a lot of reasons to think that while this is an interesting phenomenon where you're targeting a huge amount of the B cell repertoire, I really need more supporting data to know how important that actually is.
Yeah, totally. So the last thing I just want to mention, we need to wrap it up here soon, but I think regardless of the problems of this paper, I think it is interesting to think about, the fact that we can use microbes a lot of times to figure out what parts of the immune system they are trying to subvert, because that gives us insights into what parts of the immune system are actually important. And I think this is a theme that comes up over and over in immunology, is that the vast majority of microbes that we're exposed to don't cause disease. And the ones that do are specifically the ones that are able to circumvent the immune system in some way. And a lot of them have figured out different ways to do that. And I think these superantigens in particular are a really interesting way of subverting the immune system, not by eliminating some function, which is what a lot of microbial produced effectors do. They block macrophage from getting activated, or they kill T cells, or they do all kinds of other things. In this case, with a superantigen, they're actually activating the immune system, but by activating it non-specifically, they can actually hide in the overactive non-specific response. And I think that's kind of cool. Does anybody else have any final thoughts?
I also think it's cool. And I hope that I didn't come down too hard on this, because I do think that it's a fine paper. I think that, dealing with human samples like this is very difficult, and I tend to be very mechanistically driven. So yeah, you're definitely. I hope no one hears this and takes it personally, because I didn't intend it that way.
All right. Well, if there is nothing else, this has been Audiommunity episode 10. I'm Kevin Bonham with Matt Woodruff and Kate Franz, and we are on iTunes. If you didn't find us there, I have no idea how you did, but we are hopefully, as part of this revamping, we're going to actually increase our web presence in Facebook. Kate. And I'm also on Google Plus and Twitter. And next up, we're going to have a mini-sode from either Kate or Matt. I don't think we've determined which yet. The next full episode of Audiommunity will be in a month. And we hope you enjoyed this episode, and we hope you like the new format. If you do, or if you don't, please get in touch. You can find our emails and links to all of these papers, links to commentary, and hopefully some other graphics and stuff that we'll be producing at emunity.org. That's EMMUNITY.org. And the music at the beginning and the end of the episode was produced by Rachel Reinick, and she is my wife.
Kevin's wife now.
I got married recently. Not because of the music, but that's a nice side.
It was solely because of the legality of using the music. We had to sell Kevin to Rachel. It was worth it.
Yeah.
It's good music, so.
But yeah, that's it. And we're just going to go out in a sort of slow fade.
Okay, bye.
Bye.